Yeah. Oh. Okay. All right, everybody. We can go ahead and quiet down. We're going to get started. Can you hear me? All right, a little closer. Noted. Hi, everyone. Welcome. I'm Kelly. If you haven't met me already, I'm the programs coordinator here at Shoals Marine Lab. Welcome to Rock Talk. Whose first Rock Talk is this? Awesome. Sweet. Welcome. So a little bit of history about this place and what a Rock Talk is first. Shoals Marine Lab is the largest and oldest undergraduate focused marine lab in the country. And it's jointly owned by the University of New Hampshire and Cornell University, which some of you might know. Rock Talks are an old tradition on the island, and they got their name because in the early days of the lab, lectures were often conducted on rocks in the intertidal. And although we've migrated indoors, these weekly seminars continue to be a wonderful opportunity for our island community to come together and learn about new and emerging marine science research. So on island currently, we have marine parasitology and disease, where students are learning about the ecology and evolution of marine parasites. We also have applied science communications, where students are exploring the tools and techniques useful to public engagement in the scientific dialogue. Also on island is research in biology, a 2-week intensive course for students to continue independent research projects. And we're also hosting marine environmental science for high school students, where they get to learn topics ranging from plankton to marine mammals in our oceans. So for those of you who this is your first Rock Talk, this seminar is going to be about 45 minutes long, and it's going to be followed by a question and answers session where you get to ask everything you've been wondering during this lecture. And for our friends online, please use that Q&A box at the bottom of your Zoom screen. We're going to go ahead and read those questions aloud if they do come up for our audience here. And for those of you in the room, I'm going to pass around this mic at the end of the talk so everyone can hear your questions. But without further ado, I'd like to introduce Nichola Hill, who is an assistant professor at the University of Massachusetts, Boston. Nichola has their bachelor's in science from the University of New South Wales and PhD from Macquarie University, both located in Australia. She studies the evolution and ecology of infectious diseases that emerge from animal reservoirs. Her research focuses on understanding how natural and man-made processes aid the evolution of pathogens in an effort to find solutions for the control of disease in countries such as Mongolia, Egypt, Bangladesh, Costa Rica, and even Alaska. She takes a novel approach to this question, integrating tools from high throughput molecular screening and bioinformatics, and studies a diverse array of pathogens, including SARS-CoV-2, influenza A virus, cryptosporidium, thank you, Toxoplasma, and West Nile virus, and a host of taxa from birds, marine mammals, rodents, and to marsupials. So everyone, please give a warm welcome to Nichola Hill.
Oh, man, awesome. Thanks, Kelly, Merrick, and Mike, and the whole Shoals crew for hosting me today. You have just a magical island. And it was during 2015, I was a postdoc, and that was my first trip to Appledore. So it feels like where I was, wasn't me, all right? And so that was my first trip to Appledore to study gulls. And so it feels like a little bit of a homecoming. Pretty sweet to be asked to do a rock talk for you guys today. So I'm going to be talking to you about how viruses rule the planet, the spread of bird flu in seabirds, seals, and in humans as well. So my favorite organisms to study are wild birds and viruses. And I really like the challenge of studying critters that are constantly on the move. So both wild birds and viruses are also really useful indicators of the planet's health. So I need to start this talk by telling you that viruses are everywhere and that they're also great. So yeah, I really did say that. And so I hope with this talk, I can actually convince you that viruses are pretty great. So we know that viruses are not merely agents of destruction, but they are these essential components of global ecosystems. And we all know this in our heart of hearts as scientists or ecologists, that everything has its place. So why do we struggle to embrace this idea that viruses are actually really useful and important? Humans have a long history of viewing viruses as the enemy. So I'm going to give you a few reasons why you should actually love viruses. And the first is an example from the deep ocean. And we're talking about an interaction here between viruses, shown here in pink, and they're actually eating this bacteria, which is shown in blue. So I thought that this example was kind of appropriate, given that we're on an island and we're surrounded by the great North Atlantic Ocean. So I thought I would start here. So these are viruses called bacteriophage. And they look a little bit like a shuttle for a lunar space landing. In fact, I'm pretty sure that that's when NASA stole their design. These are the primary regulators of bacteria in the ocean. So we know that bacteriophage, or it's cool, we're on casual terms now, you can call them phage. These ensure that bacteria don't grow explosively and outcompete other microbes. And that's a really useful process. And they do this through what's called a rule known as kill the winner. So they will go after bacteria that are overabundant and can have the potential to deregulate a lot of these microbial ecosystems in the ocean. And by killing half of all oceanic bacteria daily, yes, daily, viruses, such as phage, they ensure that plankton can photosynthesize and produce oxygen that we breathe. And so oceans produce 50% of the planet's oxygen. And this is a process that is enabled by viruses. And this role that phage play in maintaining the stability of microbial communities in the ocean and generating oxygen means they've actually been called puppet masters of the ocean, of the microbial ecosystem in the ocean. So the role of viruses in regulating these metabolic processes that support the planet is increasingly recognized. And the abundance of viruses across ecosystems, not just oceans, but also thermal sediments and also the human gut. It's now well established that we're totally outnumbered by viruses wherever you go on the planet. So viral ecologists are increasingly motivated to quantify ecosystem processes that are triggered by viral activity. And so I had to include this really cool picture of a group of viral ecologists that are out at the deep sea and collecting different types of phage and marine viruses. And so here's this example of what we've been talking about. Ugh, apologies, hard to pick this up. But basically, we know that these marine phage are regulating these ocean processes. And in terms of like going around and eating bacteria and killing and sort of slicing their guts, they also liberate and free a lot of these nutrients that then can be consumed by other phytoplankton and zooplankton that exist in the ocean. So this basically means that they can optimize photosynthesis in that way and produce lots and lots of oxygen that we need. So the traditional microbial loop, the traditional loop of food web is on the right side, and on the left side is this microbial loop that has viruses in the middle that we're getting a newfound appreciation of. So here's a second example if oceans aren't your thing. And so this is an example from Yellowstone National Park. We know that viruses can actually form these symbiotic relationships. So here we have this continuum. Antagonism is down one end. This is when one member benefits at the expense of another. Commensalism is in the middle. So one member benefits with no consequence for another. And on the other extreme is mutualism. So all members receive these fitness benefits. But we're pretty good at really focusing on viruses as antagonists. And we forget that commensalism and mutualism is entirely possible when it comes to viruses. And a really great example of this from Yellowstone National Park is that viruses can actually help plants adapt to heat. So we know that viruses can form three-way symbiotic relationships with plants and fungi. So here we have these very large double-stranded DNA viruses that infect a specific kind of fungi that's called C. pertuberata. And viruses, so fungi that are infected with these viruses go on to colonize plants, and they show a twofold increase in the expression of an enzyme that helps the plant maintain cellular functioning under heat stress. So viruses are actually allowing plants to adapt to these hotter soils and a warming planet. And so viruses are actually playing a really important role in maintaining biodiversity in some of these extreme locations. So here's also a bit of a take home. I've given you a couple of examples, but when I teach virology, I ask my students to draw on the blackboard what would happen if all of the viruses in the world just disappeared. And they're pretty stumped, but usually they arrive at a scenario that looks a little bit like this. And it's true, we can't really prove this, but it's estimated that if all of the viruses in the world actually disappeared, the world would be a wonderful place for about a day and a half, and then we would all die. We are so dependent on viruses, and viruses play an important part in ecosystem processes, that we would be absolutely lost without them. We would not be here. So we're continuously exposed to viruses, but the majority of these interactions are actually uneventful. You don't have to worry. Viruses aren't out to get you. They colonize absolutely everything in terms of cellular life forms. And viral discovery is way, way, way behind where we are with other cellular life forms. But we know that we have approximately or a little bit over 10,000 species of viruses that have been discovered and identified. But of those, we only have about 120 species that infect humans. So basically that puts things into context. That's absolutely nothing. You do not have to be worried about viruses coming specifically for you relative to what exists in the virosphere. But boy, when they do infect humans, it can really devastate us. And that is because there are many of us. Last count, 7.8 billion on the planet. So when viruses can adapt and figure out how to infect and bind to our receptors and replicate inside us, this can be a recipe for disaster. And so, for example, what is the last pandemic that you remember? And you might not have to think too hard about this. Anybody? What you got? I'm hearing, so we're actually still in a pandemic, right? So yes, COVID, right? So we didn't have to think too hard about this. Who could forget coronavirus? We're still battling coronavirus, the cause of COVID disease in humans, of course. Or you could also not forget this fantastic GIF by Cardi B. And if you many of my students know that I always include Cardi B memes and GIFs in all of my lectures, so buckle up is all I have to say. She's fabulous, except for the mic throwing incident. Let's not talk about that. But let's go back a little bit. So we have COVID, which is what we can remember in recent history. But when we go back a little bit further, it is influenza or flu that has accounted for 5 pandemics since the start of the 20th century. And influenza is the largest and most persistent source of pandemic viruses. And I want to clarify, when I say pandemic viruses, I mean viruses that can infect humans and achieve a global distribution in humans. So flu is absolutely out there, and it's always one that we're sort of keeping our eye on. And it also happens to be my favorite virus. It's an RNA virus, and it has the fastest mutation rate out of any of the other pandemic viruses or pathogens that were on that list. It has a really large host range that also includes humans in terms of seasonal influenza. It's the reason that we all go and get our seasonal flu shot every year. Pigs, horses, cats, dogs, bats even, there are new subtypes of influenza virus that have been identified. So they have this massive host range. And the ancestral reservoir of all influenza viruses is thought to be wild birds, and specifically water birds like ducks and gulls that spend part of their life cycle in water. So water birds are these reservoir hosts for all influenza A viruses. And we know that water birds swim, but they also fly. And so their ability to migrate over these really long distances means the footprint for virus spread can be very, very big with influenza in wild birds. For example, these are a beautiful species called northern pintail. We don't get them around this part of the Atlantic, but these are actually, they breed in Alaska, and they have migrated all of the way from East Asia, where they actually winter. And so this part of these northern high latitudes attracts these vast numbers of migratory birds. Like everything basically flies north right now to have sex. And that is what we know. And we're seeing breeding birds here. And then everything migrates south again in the winter, a lot of the time for long distance migrants. That's the general rule. But they also mean that these populations are able to sort of like mix and mingle at high latitudes, and it provides this amazing vehicle for exchange of the virus, and means the virus can evolve and adapt to new hosts. And so for the way that wild birds experience low pathogenic avian influenza, for which they are this reservoir, it basically is like mild convenience. It's similar to the common cold in humans. So mild convenience for about 24 hours, and it usually hits the young ones kind of harder than the adults. If you're an adult and you're experiencing the common cold, you might even be tempted to turn up to the office that day, despite feeling a little kind of crummy, as we say in Australia, like just kind of off, not that great. But a lot of the time, you're probably just gonna like push through. Well, that was probably pre-COVID. You should probably stay at home if you're feeling a little bit off. But the idea is that it's usually mild disease that wild birds will experience when it comes to low pathogenic avian influenza. It is the same deal for wild birds, is the point. We think that low path avian influenza is subtle but cumulative in its effects. And we only recently discovered that birds infected with low path have delayed migration using satellite telemetry. But the key point is that wild birds are not dropping out of the sky with low pathogenic avian influenza. They're kind of like how you experience the common cold. You're like, oh, this sucks, but I'll get on with my life. And within 24 to 48 hours, you've cleared that infection and you're feeling peachy again. But viruses can sometimes spill over from these wildlife reservoirs that are only experiencing mild disease. So you look at them and you just wouldn't have a clue that they have been infected with this disease. And in humans, the disease looks really severe and in extreme cases can be death. So wild mammals typically serve as a lot of these zoonotic reservoirs, such as simian immunodeficiency virus or HIV from primates or SARS-CoV-2 from bats. And so this process of a virus or a pathogen, any pathogen, jumping from an animal to a human is called zoonotic transmission. With influenza, we make host jumping way easier by putting an agricultural host in the middle of this process. So influenza is unique in that wild birds are this animal reservoir, and spillover to humans only happens after evolution to a more virulent host, into more virulent forms in agricultural animals like poultry, also swine as well, these other agricultural animals. So I want to emphasize the point that the gulls that are like snacking on, and just like hanging out on top of your roofs, transmission of influenza directly from a wild bird to a human is exceedingly rare, to the point where, with the exception of this outbreak, it's almost never happened. Usually humans that are infected with avian influenza, it has to mutate into a more virulent form in agricultural animals like chickens or pigs, and then it's basically it's on its way to mammalian adaptation, and it can start to infect humans at that point. But get this, bird flu epidemiology and ecology is super complex. And so we're now understanding that poultry, mostly poultry, but some swine too, can serve as these agricultural reservoirs, and we get spilled back into wild birds in which we are also now seeing highly pathogenic avian influenza. So they now are dropping out of the sky, but this is only thanks to that mutation that was gained to the more virulent form that happened in poultry. You can see I'm like working so hard to make this land. The wild birds, wildlife are fine. We kind of screw with that process by introducing agricultural animals, sorry, G-rated. And then we get this spillback. And these more virulent forms can happen that infect both humans and wildlife at that point. So we talked a little bit about kill the winner when we were talking about phage that will feast on overabundant bacteria, right? So we've kind of created this system too, with this massive industrialization of the poultry industry. Humans have domesticated a close cousin of water birds. And so we have absolutely seen bird flu epidemics sort of really step up into the next gear with the industrialization of animal agriculture. And so you absolutely are probably hearing about huge farms where in the millions in the US of poultry having to be culled because of highly pathogenic avian influenza. And so this is this process of taking animals that tend to be genetically really homogeneous. We put them into dense conditions. Sometimes they're immunosuppressed. They're really susceptible to virus infection. And the virus will mutate to more virulent forms because it's not penalized. No one's telling it not to. What we do is we cull and we replace those animals. And so it's actually this winning evolutionary strategy for the virus to mutate to more virulent forms. So just to reinforce, this doesn't happen out in nature. This only happens in agricultural settings. So it's pretty clear that highly pathogenic avian influenza, much like what we refer to with COVID, is a virus of our own making. But I want to get back to a little bit of molecular ecology and talk about why a virus is so good at jumping between hosts in the first place. And the reason is that viruses make many mistakes during replication. So the parent virus doesn't look like the progeny virus. So that's actually a good thing if you're a virus. That's an advantage for the virus because the outcome is lots of different diverse progeny every time the virus replicates. And so it creates this huge viral diversity, and the virus is actually able to fine-tune its viral phenotype and adapt to the next host that might present itself. And it creates opportunities for the virus to spill over and jump into new hosts, just because of the fact that it's got a really sloppy proofreading polymerase during replication. But that's a winning strategy for viruses. So viruses make many mistakes. They're very mutation prone, far more than bacteria that you can see in the bottom right. We've got the x-axis here is genome size, and the y-axis is mutation rate. You can see the bacteria that are sitting down there. Or if we considered the mutation rate of eukaryotic organisms, like humans, you would have to draw that x-axis all the way out here. We have a very large genome, but a very, very small mutation rate. And you can see where viruses are by comparison, especially RNA viruses. So there's a theme here with all of these pandemic viruses. They tend to be RNA viruses, particularly single-stranded RNA viruses that exhibit these super, super high mutation rates. And so influenza is one of these single-stranded negative sense RNA viruses that sits right at the top there and is pretty hard to compete with in terms of its mutation rate and ability to evolve and be opportunistic in really dynamic environments where it's potentially there's a new host to infect in that way. But I'm someone who I'm a bit of an eco-evo person when it comes to viruses. And so I'm interested in questions like, what are the natural and anthropogenic drivers of spillover? So for me, that's a really big question that I'm kind of like stuck on. And I always like to take that approach when I'm considering how virus evolution works in natural settings like this. So I've studied bird flu for over 10 years, searching for answers to this question. Like, what are these drivers for viral spillover? And in the early days of studying high paths, that sent me to locations in Mongolia, in Egypt. I'm holding a common teal there, a beautiful species with a satellite transmitter on it. Bangladesh, we're setting up nest nets. And these were all locations 10 years ago where high path was endemic in the poultry industry. And we get this periodic transmission into wild birds. And you're like, what's going on here? But things have changed and I don't actually need to travel so far anymore. So this is me in Alaska, which is one of our important field sites for the lab, but also the North Atlantic. I really don't have to travel very far to find cases of highly pathogenic avian influenza. So something has definitely changed. And we now have a global distribution for this virus. And I want to prove that by showing you this chart. And so we know that bird flu has actually been quite a long time. Highly pathogenic forms of bird flu have been quite a long time in the making. In green are some of the biggest outbreaks in each of these 4 different geographic regions, with the first mass mortality of H5N1 in wild birds at Qinghai Lake in 2015. And in gray circles are all of these outbreaks documented by the World Organization for Animal Health. And you can see that outbreaks in Asia, so the very top, those have actually been really consistent over time, as this region is really considered an epicenter for the virus. And we've seen 2 introductions of high path avian influenza into North America. The first was in 2015, and we think it came via Alaska, via the Pacific Flyway. So we had no idea. We're sitting over here in Appledore in New England, and we're like, no, didn't get this far. The second time was in early 2022. And now we have over, I think Melba's latest estimates are over 7,000 wild bird cases in the US. And so this is a bigger outbreak. It's not going away. And it's definitely something is up. So we think that the evolution of the virus over time has changed. And we have this emergence of this new clade, and it's a little bit different from what was circulating back at the Qinghai Lake outbreak, which correlated. And since we've seen this emergence of clade 2344, sorry, that's a terrible naming convention. Blame flu biologists everywhere. I'm going to refer to it as clade 2344. But this is, we've seen this marked increase in the size and scale of high path outbreaks since the evolution of this particular lineage of the virus. And this is what it feels like to be a flu researcher, because we kind of saw this happening in slow motion. We kind of saw the evolution of this virus. We kind of saw this increasing scale and magnitude of outbreaks in wild birds. And we're like, something's coming, something's up. This just isn't right. Like, this is like red alert, red alert. We just don't really like the signals that we're getting out there in poultry and wild animals. And so for the last 10 years, we've seen it being simmering away. And now, so it's not a pandemic. I just want to say that, but it's a pandemic in wild birds. It's actually called a panzootic. So we have this global distribution of highly pathogenic avian influenza in wild birds now. And there is very serious talk of highly pathogenic avian influenza seeding another pandemic right on the tail of 3 years of the COVID pandemic. And I make no promises about what's going to happen there. We have some opinions in the lab. We can talk about it later. But the current outbreak, we know it's not a pandemic yet, but it certainly is a massive, large, big health risk. And we do see periodic human infections, usually in people that have handled poultry directly, like this case in Cambodia that had everyone on the edge of their seats a couple of months ago. It was a father and a daughter, and there was a lot of concern that the virus had actually mutated and had gained mammal to mammal or human to human transmissibility in this case. It turned out it was just a cluster infection, in that both of them had independently contact with poultry. So we're like, okay, bring it back down. We're not at a pandemic yet. But if you're not tuned into this, don't worry. You don't need to be too concerned. What you might have noticed is when you go to the grocery store and the price of eggs was kind of high, this was absolutely due to bird flu outbreaks in the US that caused a reduction in the supply chain when demand was going up. And so egg producers were like, and had to charge more for these eggs. But this has also been a disaster, and this probably speaks to a lot of people in this room. This has been a disaster for biodiversity, especially many seabird species, including northern gannets, and greater skuas, and common eider, and many of these beautiful seabird species that actually breed at these high locations, high northern latitude locations, along and also in the North Atlantic. And this was a case of northern gannets in the UK. And also right here on Appledore. So we had a smaller scale outbreak that infected 35 gull cases, or what was detected. So I make no promises that there wasn't more virus on the island. It's just that these were the cases that we were able to like sample and bring into the lab. So this was 28 of the great blackback gulls and 7 of the herring gulls. So it took out, it didn't take out, but it certainly was circulating on the island and was causing an outbreak here. And I know that it caused chaos for a lot of the bird researchers at that time. And so one of the questions that we had was like, well, how the heck did high path spread into the North Atlantic with this most recent and large outbreak? And so there's not much wild bird surveillance that's being done in these northern high latitudes, right? So let me help you get oriented. You're looking at this polar-centric view of the North Atlantic. And so we actually have Northeast Canada and the US is sort of on the left of the screen there. So you've got this polar-centric projection going on. And these are movement records that we looked at. And it basically suggests the possibility of spread via this sort of tag team of gulls that are shown here. Their distribution is shown in green, as well as shorebirds shown in yellow, ducks shown in blue, and geese that are shown more in the red. And these are species that do breed in the Arctic tundra and then they migrate along the Atlantic coast. And we know that we had our first detection in Newfoundland in Canada in December 2021 at an exhibition farm where we saw a great blackback gull and also poultry were concurrently infected at the same time. So we're like, oh, this is all we got. We really have no idea how it got here. We got nothing. We got scraps to work on in terms of like trying to figure out and how this virus actually got into North America. And so we actually used viral genomes to estimate how spread happens, to infer some of these source sync transmission dynamics that's summarized over here in this map. But you should know that this map is really pretty, but the underlying structure of this map that gives us this information is a giant phylogenetic tree. So we basically downloaded all of the high path hemagglutinin gene sequences that were available at the time between 2016 and 2022, and we just went for it. We were like, what is going on? Viruses are awesome for this, because you know how we were talking about the fact that they have a high mutation rate? So their viral genome carries all of this information about where they've been and the different hosts that they've traveled with. And so they're just prime in terms of organisms for using phylogenetic tools like this and understanding how viruses move or different ecological processes. And so we're able to sort of like on the scene of like at a crime scene, we're able to sort of like reverse engineer and figure out exactly how this happened. And we identified that Europe has become this sort of like new jumping off point for transatlantic spread into North America. That hasn't happened before. So we're kind of curious about that process. And so this analysis involved constructing a Bayesian phylogenetic generalized linear model, so a phylogenetic GLM, and evaluating all of these. The important part is that we evaluated all of these different ecological and climatic predictors for that period leading up to virus spread. And when we looked at these, it turned out the distance between the centroids was the strongest supported in terms of a predictor for virus spread at that time. So distance between centroids is just a really fancy way to say the virus, when you move closer to an outbreak, you're more likely to be downstream and to get infected. But that hadn't happened in the prior 10 years. We knew that something was circulating in Europe before we became infected in North America. So it's sort of this intuitive result that being close to the source of an outbreak carries higher risk for locations that are adjacent. Cool, got it, makes sense. And so that 2021 introduction was really unusual in moving in via Northern Europe, which had never happened before. And so we know that that was a slow process. And then once it got into the US, which became the focus of our analysis for the second part, we saw that movement between different regions in the US was incredibly fast, really, really fast. So we had movement from the mid-Atlantic and Midwest, shown here in yellow, moving into the Northern Rockies and Plains and the upper Midwest. And that occurred very, very, very quickly, consistent with how wild birds and poultry together would be able to move the virus. And we've had no evidence of demonstrated spread back to Europe from North America since that time. But we also have evidence of many, many incursions along the North Atlantic since that time. This was just the first one. So we think we have at least 2 to 3 more incursions from Europe, which I'll talk about when we get to marine mammals. So in the US, our first detection was in hunted American widgeon, another beautiful species shown here in the Carolinas. It helps that there's like strong hunting culture in these locations, often where we get our first viral detections. Like California, Texas, the Carolinas, usually that's where we're like, oh, we have bird flu because hunters are picking it up. And so after introduction, the virus spread from the Atlantic coast all the way westward and has now been detected in 49 states, including Alaska, not Hawaii. I need to double check that. And we've seen continuous circulation in the Americas since that time, raising the question, is this virus actually now endemic here? And so one of our other questions was, well, is highly pathogenic H5N1 helped or harmed by climate change? Like, why now? We know we were seeing this increased activity in Europe that's predictive of when it might come across. And so we looked at this, and when we investigated it, it turns out the temperature change at both the origin and the destination reduced spread of the virus. And so it's well known that RNA viruses are not very stable. These are enveloped RNA viruses. They degrade really quickly in the external environment. And so they just do not last for very long in sort of like these warm, humid conditions. So that kind of tracks. We're like, oh, if things are getting warmer, then maybe the virus isn't spreading as much. This makes sense in terms of understanding the viral ecology. And the way we measured temperature change was the difference between the mean temperature in 2020 relative to the difference in mean temperature from the past sort of century. And all of that information was downloaded from NOAA. So is this the strange kind of world that we live in where climate change is actually protective against the spread of zoonotic viruses that rely on movement and exchange at high latitudes where birds are breeding? And many wondered whether climate change was actually the reason why this introduction happened now. So this type of analysis actually poses way more questions than answers about climate change having an impact on bird flu. And I think this is just one of the layers, but there's way more to it. And we just know that climate change isn't that simple. So what we did with this analysis, what we completed was we took these long-term shifts in mean temperature, right? We know that we're moving from the left to the right, like things are getting warmer in terms of average temperatures. So we checked that box, we totally did that, included that as a predictor in our phylogenetic GLM. But what we didn't do, and this is the stuff that I think is like the next sort of call to action for a lot of us in this area, was try to understand rapid shifts in extreme weather. So we're not talking about a shift from left to right. We're talking about how these distributions are actually starting to flatten. And we're getting these really chonky, long tails on weather patterns. So this is extreme weather, like heat waves, cold snaps, wildfires, all this type of stuff. Didn't include that in our model. And I think a lot of people anecdotally think that that has actually driven the spread and the movement of wild birds that account for a lot of the movements of the virus from Asia into Europe, as well as Europe into North America. And we just didn't track that. So I think this is the next phase for looking at how climate change, which comes in multiple flavors, is impacting our study organism like this. So watch this space, because that will be the next iteration of how we progress with this. One of our other questions was, how do domestic versus wild species contribute to spread? We know that there's way more to this than ducks. Waterfowl, specifically dabbling ducks, are emerging as this potential super spreader of high power. in nature. And put simply, we don't get mass mortalities of dabbling ducks like we do in seabirds. So there's something up, right? And that kind of clued us into the fact that maybe it turns out that ducks are actually becoming resistant possibly to this virus. And we know that we've seen this massive host range expansion in the US in wild birds. Over 150 of our species, of the 1,000 bird species in the US, have been infected with high-path H5N1. That is a massive number. And these are monthly cumulative totals. So these pink bars will only actually increase. And we can see it's sort of starting to stabilize, which is like a bit of a good sign. So shout out to Jonathan, one of my grad students, for preparing this chart for me. We suspect that there are other super spreaders though, apart from ducks. What's going on? So I have spent a lot of time in parking lots across America. Shop and stop, got nothing. This is what I do for a living, is I go and I capture gulls in these strange locations. And we are concerned that seabirds that are dependent on human resources might start to be playing a role as super spreaders. Also geese. In terms of host abundance, geese are this prominent example of a host taxa that is successfully adapted to human activity, right? So think about like your attitudes towards birds like Canada geese. You're not alone. They have experienced exploding population numbers along with greater white-fronted geese, snow geese that are basically considered a pest in other flyways across the US. So we're kind of like, what's up? What's going on with all of these other bird taxa that we think might be playing a more prominent role in the spread of the virus? And so we used a similar approach where we used the viral genome, but this time we applied a host-specific diffusion model. So we teased it apart and we're like, well, how are different host taxa actually moving this virus? And we found that fast, efficient spread is really due to that seasonal migration of wild birds, particularly gulls and ducks. But gulls were responsible for moving high-path more rapidly than any other host. And this is a finding that may reflect their really long distance pelagic movements and also their immuno-naive status against the subtype. So they rarely come into contact with high-path, with the exception of this outbreak. Land birds like our raptors and crows, they were really among the slowest spreaders of the virus, akin to domestic birds, which don't really move the virus very much at all. We consider this is how domestic birds are moving, are just how we move poultry across the landscape. They're good to eat, and this is about just poultry trade routes. So that explains why poultry don't move the virus very far compared to all of these other wild bird taxa. What was kind of interesting is that we found that gulls spread high path actually faster than their own gull-associated low path virus. This is the stuff where they're like, this is just a common cold, it's no big deal. And they were actually able to move high path much faster than they did their own sort of gull-associated or gull-adapted viruses. And we think this is a result of the fact that they actually have strong adaptive immunity against H13, shown in green, and H16, shown in purple. So when we test these birds, they often have quite high levels of antibody against H13 and H16, but they haven't seen high path before. And so they're completely immunonaive. And this kind of jives with this idea that this first detection of high path occurred in greater blackback gulls. We know that this is a complicated story, but we think this is one part of it. I'm going to kind of, I've just seen the time, so I'm going to skip over this section where we were looking at how transmission pathways happen between wild and domestic birds. The takeaway here is that geese and swans, it turns out as a wild source, they're really impressive as a source host or to other wild bird species, including their own domestic counterparts. So this is really interesting because people haven't really looked at this before. They've just gone, wild birds are over here, domestic birds are over here. Let's run that viral phylogenetic analysis. And they haven't really gone to some of these more interesting lower taxonomic resolutions to understand how transmission pathways are going. So we think that some of our super spreader species are sort of changing. We think that ducks are here, but there are weird circumstances where gulls can play an important role, as well as geese in terms of amplifying the virus and acting as spillover hosts into poultry as well, which is kind of interesting. So this is the big one that I think everybody is here for, is what is the spillover risk to mammals? Because that's like getting closer to us, right? So one of the things we figured out is that mammals that eat birds tend to be more linked to infection. We have seen hypath move up the food web in ways that is just creepy. So many of these top predators infected with hypath, and these are just a few. So we have seals, we have dolphins, osprey, eagles, hawks, as well as foxes and coyotes, right? It's just this menagerie of different wild mammal species that are now infected with highly pathogenic avian influenza. And we absolutely think it's these food web interactions that have accounted for why all of these like top predators are getting infected. And you can see dabbling ducks in the middle, sort of down the bottom. It turns out that a lot of stuff eats dabbling ducks. They're just like way down the bottom of the food chain. So it's provided all of these opportunities for hypath to move into some of these predator species, including humans. So they're way like the turnover of dabbling ducks, like when you get band returns, you're like, wow, they just do not last in the wild. They're just, especially mallards, they just are like everything eats them. Sorry guys, like red-tailed hawks, bald eagles, humans hunt them. It's just tough to be a dabbling duck. And so they get really high turnover. And that's why we think that a lot of these food web interactions have accounted for that. But there's obviously more to it. But what about our seals? So a different transmission route we think is responsible for infecting marine mammals, and that's waterborne transmission. So mammals that swim or overlap with wild birds in these coastal areas, such as seals and dolphins and sea lions, have also become infected with highly pathogenic influenza, sometimes resulting in these really large mass mortality events. And a recent case of that is these 3,500 sea lions infected with the virus in Peru. And when we looked around, we're like, oh boy, there's 35,000 sea lions that have hypath. You know, this is making us really nervous about this virus getting starting to develop adaptations to mammalian populations. And in that vicinity, there were all of these brown pelicans and gulls and seabirds that were also dying. So the final chapter for this is that we have absolutely seen outbreaks in the Gulf of Maine. So this is like right here, super close to home. So I'm like bringing it home for these final slides. So we have seen infections in harbor and gray seals. This was work that was done by the very talented Wendy Puryear and Kate Sawatski at Tufts. And that was my former science home before moving to UMB. And they were tracking closely the virus in seabirds, so common terns, as well as common eider, a sea duck, as well as seals concurrently. And they were like, what is going on with this outbreak? And they were observing that these outbreaks were like peaking in these sea duck and seabird populations before they hit marine mammals. And then we got this huge big outbreak. So Kate and Wendy's theory is that we might have this sort of coupled system where we see infections or peaks of infection of hypath in our local gull species, as well as raptors that are probably eating some kind of duck that's low down in the food web. Sometimes we get spillover into shorebirds, and then we get into eiders, and it's like, whoa, let's put the brakes on because it's probably going to get into seals. And that proved to be the correct hypothesis last season. And this is the tail end. So this is January and February of 2023. Sorry, I don't have dates. Most of that is 2022. January, February is 2023. So we're like, is it going to happen? Are we going to see another mass mortality event in seals? So the happy news that I have for you, and I would like to finish with this slide, is that it's actually been a really quiet flu season for wildlife. We've recently seen this lull in hypath outbreaks across the US. Appledore gulls, we've been screening those. All that we've screened so far are negative, and we've seen no signs or symptoms of infection in 2023 on our Appledore gulls. No marine mammal die-offs in 2023 either. So that is indicated by this arrow. But what will the fall migration bring? I don't know. I don't have a crystal ball for this. I think this is a virus and we need to stay kind of tuned, but just be thankful for the fact that it's having a little bit of a lull, like it's having a break, bit of downtime right now, and that we haven't seen these big mass mortality events for 2 years in a row. So things are changing out there, and I'll have more to report. So thanks so much. I just wanted to acknowledge all of the folks on this slide. So many collaborators helped pitch into a lot of this work, including my team at UMass Boston. So Melba, Jonathan, and Kate that are here, right here. And if you want to talk about gulls or viruses or evolution or anything fun like that, just hit them up. They love talking about all that stuff. The Prince William Sound Science Center in Alaska, my former science home at Tufts, including Kate and Wendy that are stellar scientists. Mass Audubon, of course, Sarah and Mary Elizabeth, who work hard on the Appledore colony, USGS, the NIH, and folks at UGA. So all of those folks have pitched in and helped with this. And so thank you so much. And if you have any questions, I'm happy to take them.
Hi, I was wondering if there's any kind of interest in the risk with other domestic species, like non-agricultural, such as, is there a risk in relation to like the connection between songbirds and outdoor cat predation or anything like that?
Yep, totally. Great question. So songbird, we think that there are some phylogenetic constraints on this virus, which is kind of interesting. And songbirds kind of get infected around the edges, but we don't see really big numbers of songbirds that get infected. And this is interesting because backyard birding is massive in the US. There are lots of pairs of eyes on American robins and northern cardinals and blue jays. Like, we know our backyard birds pretty well, and we're just not seeing them die. So I think that this is happy news in that there's a phylogenetic constraint on the virus. It's not, songbirds just aren't acting like this really good susceptible host for virus replication. But your question about domestic pets is awesome, because we are seeing infections in cats and dogs, where there was recently an infection in shelter cats in South Korea. And they think that that's really concerning in terms of the virus is starting to gain the ability to not just move from a bird to a mammal, but from mammal to mammal, so cat to cat. So absolutely keep your dogs on a leash is probably my takeaway for that. When you're walking outside and in natural areas where we do have water birds, because I make no guarantees that dogs won't get infected. They absolutely do with high back. And so we have seen those spillovers happen, but it's usually when cats and dogs are predating probably more on other types of wildlife and not songbirds. That's a great question. Yeah, awesome.
What are some possible reasons why the infection rate is so low now?
$1 million question. So we don't know, but I will say there's a couple of things that we're paying attention to. Europe is also experiencing a lull in high path avian influenza activity, and we know that we are downstream of Northern Europe. And so we think that the lull in Northern Europe has meant that we're getting a quiet season. So there's just less introduction events from Europe into the North Atlantic and then across into the US. We also think that wild birds have actually, they may not have done so well. And so we're pretty concerned about wild bird numbers in terms of breeding success having reduced this year, because they experienced high rates of mortality due to the virus last year. So it may have just done this sweep through and taken out a lot of susceptible hosts. And what remains, those hosts either died or cleared the infection, and just we're seeing the core that were never exposed, or the core or the other group that gained resistance to the virus, but many got eliminated. And so it's an interesting question, because you just don't have great numbers, or it's really hard for us to quantify so much of this stuff, because it's happening on remote seabird colonies where people are just not visiting very often. So Appledore is kind of unique like that. But yeah, we don't really know. And I don't know that it's gone for forever, because we totally have low level circulation throughout the year from a lot of our samples that we get from New England. In terms, we're fairly frequently like picking up virus in raptors and weird species, but it's not like these giant peaks. So it's sort of like a stay tuned situation. But that's a great question too. Thanks.
What methods do you use to catch gulls when they're in sort of non-Appledore settings?
Oh boy. French fries. So in a lot of the parking lot stuff that we did, we were collaborating with like DCR and state agencies and using a net launcher. What's the other one that we use on Nantucket? We also use scallops. Yeah, the rocket net. So gulls are really super food oriented, and they'll cue into foods that there is sort of they're habituated to in that environment. So in parking lots, totally. Doritos, I highly recommend. French fries. When we're out on Nantucket, they're a little bit more selective, and they'll go for like scallops and things like that. So whatever they're eating in their local environment, if you put it in a bait pile, get them really early in the morning when they're a bit hangry, they'll come in, and that is how we do a lot of. But that's a great question, because it's hard to get wintering birds when they're off the nest. It's really tricky. It's a lot of work.
Hello. Thank you for the talk. And so I was curious about, since you were talking about how in a few examples, it was very similar to a common cold for some of the species. Is there anything that would cause that to become more severe? I guess I'm thinking about like climate stressors.
Okay, great question. Yes. So I think that immunosuppression is no joke, and can sometimes be this tipping point that means that a virus is actually able to replicate, and a particular individual becomes a super spreader, where before they were actually able to mount a robust antibody response and clear the virus. So those situations are definitely occurring, and we're seeing, poorer body condition, mismatch in phenotype, like birds are missing their food source. They're migrating earlier, they're spending longer on the breeding grounds. It's a big hot mess. And so we are really concerned that some of those effects directly on body condition and sort of the physiology of wild birds might play into the fact that before they were resistant, even to things like high path, like in ducks, that might tip them over the edge, and they're like, it ends not well in those circumstances. Hard to get data on it, but we're worried about it for sure. That's a great question.
How do you when you suspect a bird is has caught the flu, how do you catch it and tag it or whatever without having you getting the flu with you and then moving it to other species?
That is such a great question. So I think anyone working in emerging infectious disease has to be really just constantly thinking about how to minimize the infection risk to yourself, but also to other species, and carrying the virus even in fomites and around on your shoes. It's not easy. So we always take really major universal precautions whenever we're in the field, and so we're wearing a lot of PPE. And what else do we do? Masks, gloves, everything gets destroyed, put in biohazard. But yeah, those are situations where probably when you're in the middle of an outbreak, it's probably kind of too late as well, for that particular population. You could probably play a small role in moving a virus to a completely new situation. But I think in those local circumstances, it's probably transmission is taking care of itself. But I think it's a great question. We are always really, really taking major priorities to make sure and major precautions to make sure there is no spread that's caused by researchers. So I think on Appledore last year, correct me if I'm wrong, when they were finding dead birds that were positive for hypath, there was like a boot wash station. I think a lot of the colonies with seabird researchers, people are wearing Tyvek suits and like burning them, and just it's a massive, it's been like a huge consideration for anyone that's out there on these colonies. I don't know, does anybody remember Appledore? I wasn't here last year. Yeah, do you remember it? What happened? Yes. Yes. Yep. Yeah. Yeah. Right. Mm. Mm. Ooh. I like how you like flipped that question back on me. Yeah, we can test that in the lab. Yeah, you know, I think so. What I'm hearing is that the best approach is, I think all of those things are correct. I've heard of people wearing booties that they basically just dispose of at the end of the day, always gloves, always masks as ways to protect yourself. And here's the other interesting part about reducing spread on these seabird colonies that are just being devastated, is to actually remove carcasses. So people are seeing, and they've done tests on this, whenever they move dead birds off the site and incinerate or compost, it can actually really help reduce the spread of the virus in those locations. So that kind of, I mean, you obviously need to have someone who's got a Tyvek suit on and gloves, and they're moving it off for you with all of the full PPE, but actually that's really helpful. So it depends how much money you can spend on PPE, unfortunately. But I think as entry level, I think that was like a great solution for minimizing spread. Yeah, yeah. Mm. Yeah. Oh yeah. Yes. Yep. Don't kiss your chickens after you've been to Appledore. Yeah, I would honestly like put myself, if it was me, I would put myself through my own like 7-day quarantine, where if I'm in an outbreak on Appledore, I probably wouldn't interact with poultry. I would have somebody else deal with the husbandry of the poultry for like 7 days, and then can start interacting with them again. I think the risk from wild birds to humans, I think we're sort of touching on that before, is very low, but I think humans can, we don't have great data on it, but could potentially be playing a role in like moving it around. But yeah, it's really a tricky one. I think best practice is like the boots and the gloves and the masks, and trying to sort of remove your PPE at the end of the day is probably the best that we can do. But it's hard. I mean, you go outside your door and there's like, you know, herring gulls nesting right outside of the residences. And so there's just birds everywhere. So it's really a tricky one. I wish I had some better answers for that. Yeah.